And so now we have to move on to our next speaker, who is known to many of us and who's been the balik ng balik Filipino scientist for many, many decades. He's an expert in marine drug discovery and development, distinguished professor of biology of the University of Utah. He is the first Filipino elected in the U.S. National Academy of Science and also a member of the American Philosophical Society. Our speaker holds over 300 publications on peptides from marine cone snails and their specific molecular targets, considered as founder of the field of conotoxin pharmacology. Let us all give a round of applause to Dr. Baldomero Toto Olivera.
It's always a great pleasure to be here. I think maybe it wasn't a coincidence that the first three speakers in this panel are all graduates of the Department of Chemistry in UP. So we are very grateful for that heritage. I'm going to try to do something maybe a little different. What I'd like to do is to be more prospective than talk about stuff that has already been done. I will talk about some of the stuff that's been done, but the theme I'd like to develop is what can PAASE do in the field of biomedical science. And I was very happy to hear the Dean of the College of Science saying that his mission is to bring together departments, because in fact today that's more important than ever. And in order to kind of give you an idea of where I'm coming from, I think 10 years from now, the field of biomedical science will be completely different and will undergo a revolution that's even more far-reaching than the recombinant DNA revolution of 20 years ago. And what I'd like to do is to suggest that the Philippines can place itself in that revolution so that instead of catching up by taking advantage of certain very important features of the Philippines, we can extract science that nobody else can do. And we may find ourselves, I think if we play our cards right, not trying to catch up with Vietnam, but in fact being ahead of any other country in several respects. And so what I'd like to do is go through this diagram. So traditionally, if you want to do something in biomedical science, you start with basic science, and then the biotech industry, the biotechnology sector, then converts the basic science into biomedical applications. And so that, of course, is the traditional way to go. And what I'd like to do is to go through what our own experience is. So many years ago, Luli Cruz and I decided to work on this Philippine snail, Conus geographus, and the young people in the audience always want to sort of find out what our vision was, but the truth of the matter was at the time we had nothing else to do. And the reason was we had no equipment at all, and so we had to decide to do something. And we decided to work on the snail because the snail killed people, and we knew that it was venomous. And in fact, the way the snail hunts fish is something that we learned by having live snails. And this is a video that shows you the strategy of this particular snail. As it approaches the fish, it opens its mouth, and it's releasing venom components that are changing the behavior of the fish. And so what's remarkable is that the fish doesn't swim away. It knows something is happening, But as the snail approaches the fish somehow never swims away, and it allows the snail to completely engulf it. And that's because the venom components that the snail is releasing are having an effect on the nervous system of the fish and are knocking out the sensory perception of the fish. And after the fish is engulfed, it's then stung by the snail, venom is injected, and this fish becomes paralyzed, and the snail is able to enjoy a big meal. So this is what we observed the snail was doing. And at the time, let me start with this first. What we knew was essentially nothing, okay? But today, thanks to basic research, we know a lot more about this particular snail. And there are lots of scientific publications. And in fact, from this basic research, there are at least 10 biotech and pharma companies that have looked at compounds from the snail. And as a result of that, we've ended up with an approved drug for pain and several leads. And what I'd like to show you is, that this all came from the study of that one snail. And so what you can see is that there's one approved drug called Prialt. There are two that are in human clinical trials, one for epilepsy, one for pain. And there are two that I've listed here, including a preclinical candidate for diabetes. So that all came from a single snail. And what I'd like to do is to convince you that by studying basic science and combining biology and chemistry, that you have completely unforeseen results. And that certainly was the case for us. So that was the beginning, and now we're just starting to try to increase the biomedical applications. But what's the situation in the Philippines? Well, the situation in the Philippines is that the basic science is now really in great shape. We have knowledge of the biology and the chemistry, and the clinical end also has good potential. We have great clinicians. And the challenge for us in PAASE and everybody involved is that the biotechnology sector, the endogenous biotechnology sector, is essentially non-existent. So what I'd like to do today is just point out where we could take advantage of, in fact, basic research done in the Philippines and explore how we can make this transition from the great basic science that we increasingly have to the clinical development by essentially trying lots of ideas on what kind of biotechnology we need. So what I'd like to do is to give you my overall message, because I may run out of time. But in the field of biomedical science, because there's a revolution that's about to happen, I think that we should do the following. The big message is be local. Take advantage of the local resources we have. The Philippines is an archipelago. Each island, especially in the marine environment, has unique biodiversity that's found nowhere else in the world. However, this biodiversity, I would suggest, is enhanced by the fact that we have a hidden multicultural heritage. I think that the problem is that the colonial periods in the Philippines all focused us on becoming a single nation. But in fact, the Philippines is a multicultural place. There are over 70 languages. And in every island in the Philippines, people know a lot about their biodiversity. That is scientific knowledge that we should start with. And I think if we are able to inject a little bit of science and essentially take advantage of this biodiversity and our cultural heritage, there will be an incredible synergy which allows us to, given what's going to happen in the biological sciences in the next few years, allow us to jumpstart and be in a different place as far as biomedical science is concerned. So let me go over my ideas very quickly. I think that PAASE and PAASE members should be able to take advantage of what we've taken advantage of, which is that we can take compounds that we already know about and perhaps convert them into industry, biomedical industry. And so I'll take examples from our own work. From Conus geographus, we know there's a compound of the venom. The venom has 200 bioactive compounds in that one species alone. And one of the most notable compounds, which of course Luli and I gave a Filipino name to, is called contulakin. Because when you inject it into mice, you have to poke the mice before they run up. So normally you just tap a mouse with a pencil and it runs away. But the mice injected with this peptide have to be kind of really prodded. And then they get up and they sit down again. And so we call that peptide contulakin. There was another peptide that we found at the same time that put mice to sleep, and we called that conantukin. So contulakin and conantukin are very important peptides because they've both reached human clinical trials. And what we hope to do is to push these so that our clinicians can use them. And contulakin in particular is an interesting compound because it resembles a neuropeptide called neurotensin. And what it does that we found is that, like no other compound known, it's able to alleviate cancer pain in advanced bone cancer. So one of the really intractable conditions that, in fact, cancer doctors face is that, especially in metastatic cancer, there are very few options. You can give opioids, and you can give the peptide that's already approved, that we developed. But this peptide is better, and especially for cancer pain. What my collaborator has found is that in an animal model, he's never found anything better. And so this is in phase 2 clinical trials. And the trouble with the modern pharmacological industry is, unless they think they can sell $1 billion a year, they don't want to develop it. But if it relieves pain of people with intractable bone cancer, I think we should develop it. And I would really very much like to discuss ideas on how we can take a compound found in a Philippine snail, developed by Filipinos, and make it a clinical candidate for intractable pain. However, there are other compounds that are in earlier preclinical development. So when the snail approaches a fish, one of the reasons the fish doesn't swim away is because the snail is releasing insulin. But it's the smallest insulin that's ever been found. And as a result, the fish becomes hypoglycemic. And so it just sits there without any energy. And that allows the snail to essentially engulf it. And what we found is that the snail has designed an insulin that's the fastest acting insulin that's ever been found. And so, although this compound was only discovered 1.5 years ago, we now have a patent on it, and our collaborator has put some of the features of this insulin into human insulin, and there is a biotech company that wants to develop it as a preclinical trial. So there's a lot of opportunity from the basic research that have clinical possibilities. And I'd like to highlight one of the most recent ones done by Bea Ramiro in Giselle Concepcion's laboratory, because what she found was that from a cone snail venom, when you injected this into mice, the mice suddenly became very quiet and after a while non-responsive. And what we found is that this is in fact a compound that alleviates pain. And so we're just patenting this, and the people at the University of Utah recognize that in fact the contribution of the Philippines was huge, and so they asked me to estimate how to divide the IP, and I said, well, 50% should go to Bea Ramiro, because in fact she did all the work, and then Giselle and I and Lita Imperial sort of kibitzed and watched, so perhaps we should get the other 50%. And that works out to 2/3 for the Philippines, 1/3 for the University of Utah. So I think that this is a model in which now we have IP, we have a compound from a Philippine snail, and we have a non-opioid acting pain medication. And so we should take advantage of that. And I'm pretty sure that people in this room might know how to set up the right type of biopharmacological or biotechnology company that we can take advantage of. So we also have a program of antibiotics and antimalarials that Giselle has developed in collaboration with some of the people at the University of Utah. This just shows you what that little insulin looks like. And the reason it works so fast is because insulin that our pancreas produces is a hexamer. The hexamer has to dissociate to a dimer and a monomer, and it's only the monomer that passes through the capillary wall. But the snail has designed this insulin so it's made as a monomer, and that's why it's so fast acting. So here are the people who've contributed to this work, but I'd like to take 2 more minutes, if I may, to just show you what we don't know. In other words, I've told you about compounds. There are 200 compounds in Conus geographus, and I've told you about 3 of them. But recently, we discovered our ignorance was greater than we thought. So here's Conus geographus. We put 5 fish in the aquarium. 1 disappeared, and the first one disappeared. You see it's swimming funny because it swam straight for the mouth of the snail. And so you can see, we thought, stupid fish, no wonder it got caught. But then, what really blew our minds was the second event, because we had a camera on the whole night. The second event, which we call the last pirouette, this fish, the snail, has secreted something that makes the fish swim really, really weird. And so that's impressive in itself. But if you watch this video, what's most impressive of all, that snail is just sitting there. But you can see it's moving a little bit. Okay, watch the fish. The fish swims straight for the mouth of the snail. Boom. The snail is doing this chemically. It's secreting compounds that change the behavior of the snail so that the snail can be captured. This is the same snail that went straight for the big fish. And so what this tells us is that these snails are capable of much more amazing things than Luli and I ever realized when we first put our first 3 Conus geographus in an aquarium. And for the young people, I would say that the Philippines is so rich in biodiversity, you don't have to work on venomous snails. You can work on these guys, the vampires of the tropical seas. They haven't been studied at all. And what I'm going to show you is how they operate. They extend their proboscis, and now they're going to attach the proboscis, and these guys will eat nothing except blood, the blood of a fish. And what we're going to show you is a gruesome technicolor afterwards, and you can see the blood, and there it is. And we have found that the snail seems to be using a completely different strategy from leeches and from vampire bats. And in this case, the fish was rather small, and so in fact, I won't show you the more gruesome part of this, but you can see they have to have incredible anti-blood clotting factors. And just so you remember this, I'm going to haunt your dreams by showing you a large fish and 4 snails sucking blood. And we know nothing about the snail. You could work on this. It's found nowhere else except in the Philippines. And so this is the kind of project that I think we can make the basis for our future, hopefully very advanced biomedical industry 10 years from now. And so I think I would be remiss if I did not really say that the real heroes are the people who hunt these snails. And because the Philippines has a maritime culture, and because the Philippines has a lot of people who know the biodiversity, where to find it, how to harvest it, that is a scientific resource that PAASE should begin to try to think to develop. Thank you very much for your attention.
Wonderful. Thank you. So, Toto Olivera is tireless, ageless, and you can understand why. He has this passion for trying to discover the mysteries of nature. So much to learn from nature and from evolution through Toto Olivera. So thank you so much.